Rahman Rahim, we start the part 2 of the paper that I've just previously uploaded. Is a paper 4.2 solved March 16, paper 4.2, and from here we continue with the question 4, which I left halfway in the previous video. Starting with question 4, the process of gametogenesis in male and female vertebrates, including humans, involves meiosis. You describe how gametogenesis differs between the human male and the female, and I just drew a line in between and I'm giving you the differences. You can do that. There's no way that it doesn't say anywhere that you can't draw a line. So female male begins before birth. In females it begins before birth and male begins at puberty. Then in the female one ovum is released every month, and in the male four spermatids are produced from that one spermatogonia. And then meiosis is interrupted because the woman has these in the at birth, but then she doesn't have. So meiosis is interrupted, and this is a continuous process. And in the male, there are greater number of gametes, and in the female, it's produced gametes to a greater age. Part of the question starts with figure 4.1 shows a dragon, whatever the name is. This species of lizard is only found on 5 Indonesian islands. Read the question carefully, comprehend 5 Indonesian islands. It's a dragon. The picture is given to you, and it's a lizard. Then in 2006, two captive female Komodo dragons A and B. So these are the parents in British zoos. Each produce healthy offspring 1 to 4. So four offspring 1, 2, 3, 4 produced by two captive females A and B, despite never having mated with a male. Female B was later mated with a male C. So B was then mated with a C, and they had an offspring called 5. Now do all this horror work on the paper and explain this paper to yourself. The results of the genetic analysis are shown in table 4.1. Different alleles at 4 loci. So we have gene locus P, Q, R, and S. So we have 4 loci, P, Q, R, and S, and could be distinguished by different lengths. Now, what are the lengths? Allele length is base pairs. Please understand what do we mean by allele length. Like for instance, if I say AT, GC, now these are 4 base pairs. But if I add another one here and I say, okay, so this is 5 base pairs. 5 base pairs. So base pairs is the length. So that is called BP base pair. So this is now 1, 2, 3, 4, 5 base pairs. Now the gene loci tell you this is 211 base pairs. This is 216 base pairs. So that means there is slight difference in length. And that means that this could be another allele. Like remember in blood groups we have IA and IO. So the length of IA could be different and the length of IA could be different. So it could be a different allele. So please first understand the questions, go back to your chapter on genetics, rethink your chapter on genetics. Now again reading through this, the results of the genetic analysis. The results of the genetic analysis are shown. Different alleles at P, Q, R, and S could be distinguished by their different lengths. The alleles for each gene are shaded differently in table 4.1, and the sex chromosomes of this species are called W and Z. So just like in humans we have XX and XY, so here they have said is W and Z. So this would be one having W and Z and one having WW or ZZ. So this is of course the possibilities that, so in animals you've got to think of the different possibilities. Now as we go on to this table, now this table shows you very interesting things which you need to comprehend. Number one, female A. So I'd read through this as I would say, okay, female A, female B, these are all the parents. So I would circle them differently. And then it says, now female A had two offsprings. Offspring from female A was 1 and the other was 2. Then the offspring from female B was 3 and 4. So I would read this, I would do this drawing like I'm doing now. I would expect you all to do this as well. And then we look at the sex chromosomes. Now female Z was WZ, and the offsprings from her were all the ZZ. And then we had female W was also WZ, and her offsprings were also ZZ. And then we had male C. Now male C was ZZ. Now what did we do? Now, if you go back at the question, we had female B was crossed with C. So B was crossed with C. So here we have this B was crossed with C, and then we had an offspring from B and C. So please try to understand this, and that is why we had, so B was WZ and C was ZZ, and then we had WZ. So this is what we had to connect, this one with this one, and this is the offspring. So read the question, do this carefully, understand the question, and then I think everything will fall in place. Then the part 1 of the question says, using table 4.1, identify the gene locus that shows the most genetic variability and name the process that gives rise to different lengths of the allele. You see, um, I was teaching this to some students yesterday, and they said, Miss, how would you know that? And I said, well, because you see, if there is a change in the DNA, alleles means a change in the DNA. And if there is a change in the DNA, that has to be something to do with mutation, because a change in the DNA has to be mutation. So the process had to be mutation. But now, which, it says identify the gene locus. So it either had to be P, Q, or an R, and S. Which one was genetically variable? Now, let's look at the previous diagram and then figure this out. Now, if you look at this table, you can see how I have given you the genetic variability. That's what they asked in this question. They asked you, using table 4.1, identify the gene locus that shows the most genetic variability. So the word was genetic variability. Now, if we go back onto this table, you see these are the ones which are different, 211, 216. So this one had 211 base pairs, this had 216 base pairs. Then again 211, 213, then 211, 216, 211, 216. So there were three of them. This one and this one were all the same, and only one was, so we have two different variations. But in R, if you look at R now, that is interesting, is 188 200, and then 188 206, and then 190 206. So this had the highest genetic variability. So this is how you read through a question, this is how you try and understand the question, and then put your knowledge to this. These 2 hours are going to be your final exam, in which you're going to really put all the information that you have from your previous biology classes or your self-study, and put it all into practice and see how you're going to figure this out. And many students did figure this out, because as I was going through the exam report, it says many students were able to identify the locus R. Locus R. Then using table 4.1 again, state and explain which animals are heterozygous at one or more of the gene loci. So heterozygous, is a capital T or a small t, or a capital A and a small a. So of course the base sequences would vary if there was an allele. But if there was a base sequence of the same, then it would mean they were both the same. So it would be homozygous dominant, or it could be homozygous recessive. So this is how we were going to figure this out. It says which animals are heterozygous. Now look at it how I've done this. Is, you see, female A was heterozygous at P, Q, and R. Heterozygous. Then female B was heterozygous at P and at S. And then male C was heterozygous at P, Q, and R. And then offspring 5 was heterozygous at P and at R. So this is how you have to figure this part of the question. Now looking at the question, you can tell that these are heterozygous, because this was 211 and 216. This was 211, 216. This was 190 and 206. So that means these are heterozygous. The others were all homozygous. So this is how you have to figure it out. Please pause here, pause the video and have a look at it, and then understand it, what I've written in the mark scheme. Part 3 of the question was again very challenging. Clones are organisms or cells that are genetically identical. Students made suggestions about the dragon offspring 1 to 4 that produced by a female that had never mated with a male. So offspring 1 and 2 are clones of each other, and 3 and 4 are clones of each other. So they want you to figure out, are 1 and 2 clones of each other, and are 3 and 4 clones of each other? Offspring 1 and 4 were produced by asexual reproduction using mitosis only. Then asked you to compare. It said offsprings 1 and 2 are clones of each other, and offspring 3 and 4 are clones of each other. So if you look at offspring 1 and 2, how can they be clones of each other? They're not clones of each other. Why are they not clones of each other? Because if you look at the different loci, if you understand the question, it says, look, how are they different? 1 and 2 are different at loci P, Q, and R. At P, look at it, it's 216, 216, 211, 211. Then at Q, it's 151, 151, and 154. So these two are different. This is different, this is different. And then at R, this is 200, 200, and this is 180, 188, 188. So offspring 1 and 2 are different at P, Q, and R. And then offspring 3 and 4, how are they different? They are different also at P and S. So 3 and 4 are different at P and S here. So this is different and this is different. So offspring 3 and 4 are different at loci P and S. So this is how you had to do that part of the question. The question said offspring 1 and 4 were produced by asexual reproduction using mitosis only. Now that is not also, so it was not asexual reproduction and mitosis only. And the reason it's not just saying not asexual and mitosis would give you an answer. No, it won't give you any marks. Why? Because 1 and 2 were different at A were different to A. Because if you look at this one, 1 and 2 were the offsprings of 1 and 2 were the offsprings of A. So 1 and 2 were different at loci P, Q, and R with comparing it with A, different at A at loci P, Q, and R. What you had to do was compare the female A with offspring. So female A was the parent and offspring 1 and 2 were there. So if we say they've been produced by mitosis, then all of these should have been the same. So this is female A, mitosis, this should same. But now what do we have? We say offspring 1 and 2 different to A, 1 and 2 different to A, 1 and 2 different to A at loci P, Q, and R. So please understand this, what we are saying in the question. So the female A was different 1 and 2. So we say offspring 1 and 2 different at loci P, Q, and R. And then of course we come up with the next suggestion. The suggestion was that offspring 3 and 4, you see these are the offsprings 3 and 4 of the female B. So offspring 3 and 4 were different to B. They were different to B, the parent, at loci P and S. You see, if this is the parent, then all of them, like in this situation, which is in which they're all the same. In this, they're all the same. So please understand this, how we are saying that offspring 3 and 4 different to B, different to B at loci P and S. I hope that makes sense and you can understand this part of the question. And that is why, what did we write here? We said they're not clones because 1 and 2 differ at PQR, 3 and 4 differ at P and S. And then we said there's not asexual in mitosis because 1 and 2 different to A at PQR and 3 and 4 different at P and S. Please pause the video and try to understand this at this point. Then the next part of the question was very theoretical. It was a 4-mark question. It says reproduction without a male has evolved in a number of species that live on islands. Suggest advantages and disadvantages of this type of reproduction in an island habitat. You know, in an island habitat, naturally there's not going to be a lot of influx. There's not going to be a lot of immigration and emigration. So advantages: males may be scarce, females can still reproduce to continue the species. Offsprings are all male, so female could mate with the offsprings. and retains the adaptations for that environment. Then the disadvantages, of course, are also there. It's a very small gene pool, decreased heterozygosity, harmful recessive alleles may come together, lack of hybrid vigor, and cannot adapt to the changing environment. To question 5. And the A part says figure 5.1 outlines how two hormones A and B are involved in the regulation of blood glucose, and immediately what should come to your mind is insulin and glucagon. Insulin is released when the blood glucose is high. Glucagon is when the glucose is gone. Yeah, the glucose decreases in the blood, so decreased blood glucose concentration. So normal rise in blood glucose, fall, rise, fall, and then hormone A produced, hormone B produced, target liver release of glucose, absorption of glucose. Now, with reference to 5.1, name the control mechanism that regulates blood glucose concentration, negative feedback. Hormone A was glucagon. And then it says describe and suggest an explanation for the changes in the activity of the enzyme glycogen synthetase and glycogen phosphorylase. So blood glucose concentration rises, insulin released, more glucose enters the liver cells, increased activity of glycogen synthetase. Naturally, the word glycogen synthetase means synthesis. So more glycogen being formed because your blood glucose is high, so it's being absorbed and it's been taken away from the blood and is being stored in the liver as glycogen. Glycogenesis is the name given to the word. Formation. Genesis means formation or production. So glycogenesis, the word lysis is used in biology a lot as well. And that's the breakdown. Like we said, glycolysis. So in the glucose breaks into the 2,3 carbon in glycolysis. So here, of course, we're saying of glycogenesis, then reduced glycogenolysis. So less of the glycogen breaking down and decrease in the activity of glycogen phosphorylase, which is required for the formation of glycogen. Now the question 6 was on paramecium, which is a ciliated unicellular protoctist. So number one, you have to realize protoctist, so it's a eukaryote. The cilia are similar in structure to those found in the trachea of a human. The cilia beat to move the paramecium through the water in which it lives. Now figure 6.1 shows a paramecium. Paramecium has anterior and posterior ends. Generally the cilia beat so that the organism is moved forwards. Sometimes reverse movement is needed, for example, when the paramecium meets an obstacle. So it needs to go forward and it needs to go back, just like in our car we have a forward and we have a reverse gear. The direction of beating of the cilia is linked to the difference in concentration of calcium ions inside and outside the cell. There is usually a higher concentration of calcium ions outside than inside the cell. When paramecium touches an object, its cell surface membrane becomes deformed. The membrane potential becomes more positive inside the cell. So we have the paramecium, and inside the cell it becomes more positive. So more positive inside the cell. The organism moves backwards for a short time. Now it says suggest the sequence of events that occur to cause the paramecium to move backwards when it touches an object. Now you have to get all this from the question, nothing from anything from outside. Calcium ion channels open, calcium ions diffuse down a concentration gradient, and cilia beat in the opposite direction. Cilia beat in the opposite direction. So everything was taken from the question. But you had to read the question carefully and you have to understand it, and then you have to put it into words. So this is the biggest exam skill of doing a paper 4. Then of course it came on to another part of the thing. Suggest how paramecium ensures that there is usually a higher concentration of calcium ions in the surrounding water than inside the cell. So active transport is we're going to have active transport of there must be a pump, and then calcium is going to be pumped out against the concentration gradient, and if it's an active transport it uses ATP. Now all of you knew that active transport requires ATP, but the majority would not have written using ATP. So even if you said active transport using ATP, it was 2 marks, you would have got your 2 out of 2. Okay. Now it's another situation. Let's look at the new situation. Read the question carefully. Paramecium has a contractile vacuole that fills up with water. When it is full, the contractile vacuole contracts. When it is full, it contracts to expel the water, and the rate of contraction of the vacuole depends on the water potential of the surrounding water. Name the process by which water enters the paramecium. Then it says suggest the relationship between the rate of contraction of the contractile vacuole and the water potential of the surrounding water. Naturally, it is saying to you in the question that it fills up with water and then it contracts to expel the water. So the rate of contraction depends on the water potential. So the higher the water potential outside, so it's distal water outside. So water is going to move in, contractile vacuole is going to fill up, and there's going to be a faster rate of contraction. Again, you have to just pick it up from the question. No, often my students come to me and say, Miss, the paper was nothing of what you taught us. It was a very general paper in which it was testing your knowledge of biology, not any direct questions. So suggest the relationship between the rate of contraction of the contractile vacuole and the water potential. So naturally in the question it has said that when it is full, so higher the water potential outside, more water will be pumped in and the contractile vacuole have to work more. Then of course comes the last part, describe how the DNA of paramecium differs from that of a prokaryotic cell. Now I told you in the beginning paramecium is a eukaryote. So eukaryotes will have a linear DNA, then they are associated with histones, then they are contained in the nucleus, and DNA is present in the mitochondria. Now this would be somebody would not remember that this is a eukaryote, so they have extra nuclear DNA as well. You know, DNA is present in the nucleus, DNA is present in the mitochondria, and DNA is present in the chloroplast. So there is called extra nuclear DNA. Then now let's look at question 7a. Figure 6.1 is a diagram of a section through a mitochondrion, and we have circular DNA, and then we have labeled P, which is the outer membrane, and Q is the intermembrane space, and R is the inner membrane, which is the folding, which is the cristae, and S is of course an M mitochondria. M would be the matrix. As I tell you often, M mitochondria, M matrix. So just to remember it, please do not confuse. In the chloroplast, the inside liquid is called the stroma, chloroplast, and the last two words of chloroplast is ST. So ST for stroma. Do not mix this with the matrix. Using figure 7.1 state the letter which indicates the site of the Krebs cycle. Krebs cycle takes place in the matrix, so S. Oxidative phosphorylation takes place on the inner membrane, so that would be R. And the decarboxylation is a Krebs cycle because carbon dioxide or decarboxylation or decarboxylase is in the Krebs cycle, and that is also in the matrix of the mitochondrion. And then of course we come on to the B part, suggest one function of for the circular DNA. Circular DNA makes mitochondrial proteins, mitochondrial enzymes. It is important in mitochondrial replication. And of course, it also makes the ribosomal RNA for the 70S ribosomes which are present inside the mitochondria. Now that somebody would not have thought of, and I can think of them asking you a suggest question on that in this year. Then it says during respiration, exchange of substances takes place between the cytoplasm and the mitochondria. Complete the table below to list 3 substances that enter and 3 that leave the mitochondria. So those things which can be enter, of course, are about, we have a list of 5. Oxygen can enter, pyruvate, ADP, phosphate, and fatty acids broken down into 2-acetyl-CoA. And leave the mitochondria is carbon dioxide, ATP, and water. Then the next part is examining another very important part of the syllabus. It says the poison cyanide binds with cytochrome oxidase, one of the carriers in the electron transport system. Suggest how ingestion of cyanide by humans stops or leads to the death by muscle failure. So ETC stops Krebs cycle stops, no proton gradient setup, no proton flow through ATP synthase, no or less ATP produced. And then, of course, they've asked you a question on the RQ. Tripart palmitin is a triglyceride. The chemical location of this is this. Calculate the RQ value. You know RQ is CO2 over O2. So, I'm sure you all know that it is the volume of CO2 over the volume of O2. So here we have 102, here we have 145, and that is 0.70. Then, of course, the last part, explain why the usual RQ value for respiration is humans between 0.7 and 1. Now, that for carbohydrates it is 1, for proteins it is 0.9, and for fats it is 0.7. And I tell you to remember it is 6 over 6 glucose and P for protein, and you just if you make a mirror image of it, P it becomes 9, and F if you make a mirror image it becomes 0.7. So they're all less than 1. So whenever it's less than 1, it is aerobic respiration. But if it's more than 1, then it's anaerobic respiration. So respire aerobically, mixture of substrates, and different tissues respire different substrates. The brain can only use glucose. So that is what we had to understand. The last question of section A, in a species of snail, shell color is controlled by a gene with 3 alleles. So allele CB codes for brown, B brown, and P codes for pink. And so C raised to the power B and C raised to the power P codes for a pink shell, and C codes for a yellow shell. Now CB, brown is dominant to both the pink and the yellow. So brown is dominant, so B is the brown one, is dominant to both the pink and the yellow, and CP, the pink, is dominant to the yellow. Now the shells of this snail may be banded, have dark stripes, or non-banded. Now the allele for non-banded is N. So non-bandeds will be big N, or big N, small n. And the allele for non-N is dominant, and the allele for banded, so all the ones which are banded, now we know this. Now this is always what I tell you, is when you bring genetic questions, is first look at the recessive trait. And that is the biggest clue in genetics questions, is to figure out the recessive trait, and then to say, okay, well, the dominant traits now can be either this or this. So state what is meant by the term dominant allele, the direct question, allele expressed in the heterozygote, and allele one or more, what is the definition of allele? One or more nucleotide sequences at a single gene locus. I think that's a very good definition. And then it says a cross between a brown non-banded and a pink non-banded. So if they're both non-banded, then of course we had this. A snail produced some offspring that are yellow and banded. Now the reason if it was banded, so we couldn't have had this, because as I've just told you that the banded was small n. So if we had this, then we couldn't get these. Then we couldn't have got these offsprings. So that gives you a clue that one of this had to be heterozygous in order to have an offspring with this genotype. So that is why both of them have to be this. And then of course state the genotypes of both parents CB, CY, and pink non-banded. Then they list the parental gametes. Well, I've told you always the gametes is a very simple way of doing it. 1, 3, then 1, 4, and then 2, 3, and then 2, 4. So this is CBN, and you got all these. I'm not going to go into the details of that. Now the pink non-banded. Then state the genotype of the offspring that are both yellow and banded. So this was the genotype CY, small n. And proportion, of course, you'd have to do this genetic diagram and figure this out. So please do this paper, and I wish you all the luck and best of luck for your paper 4. And thank you very much.